And let me introduce our next speaker, scientific, the scientist AIRI, Fishman. Veniamin is going to present the results on developing the models which forecast epigenetic changes in different tissues and organs based and using AI.
Вениамин, приветствую тебя.
Benjamin, greetings. You can
Здравствуйте, коллеги. Спасибо большое за возможность выступить на такой замечательной конференции. И на самом деле я занимаюсь генетикой, я Института искусственного интеллекта и ЦИГа. И как генетик я хочу рассказать вам о том, как можно использовать искусственный интеллект для того, чтобы решать проблемы.
I'll tell you how the AI can be applied to resolving the problems of the medical genetics. And for an instant, I want all of you to fancy yourselves a genetic doctor. Like you sit in your office and all of a sudden a patient walks in. It must be probably a small kid with congenital diseases that they manifest themselves right after birth, shortly after birth. They have some retarded patterns of development or some disorders about bodily proportions or face features or any other anomalies. And the first thing is that you should diagnose what happened and then provide some therapy. And there are very scarce options for that, and you today have around 10,000 described genome disorders that can cause anything similar to what you will be seeing, and you sequestrate the DNA and try to find a letter or several letters in genome that have caused a breach that manifests itself so clearly. Let's look at genome. Each person has 3 billion letters, and each person has around 1 million differences from others, and these letters quite often don't have to do with genes, or like the flaws may not be about genes, like those sequences coding proteins account for 1.5 or 2% of our DNA only. And why do we need the remaining 98%? There is the genetic sequence or code that is a set of rules that defines how the changes in the genetic sequences will impact the protein sequences. But that is only about 2%. But what about the rest 98% of the letters? And what is to be done about these 98%? No one knows, because there is no simple code or set of rules. And how, like, changing this letter, what effect would changing a letter have? There is a plethora of different mechanisms, and each year we discover new ones. Let me show an illustration and an image from a Nature article. It says that if you change the letters in the sequence, that has to do nothing with the gene itself, but is next to it, that will entail a transformation of the cell into a malign one, into a cancer cell. As this sequence was used to form and shape loops, and if you change that, the loops would also change shape and the gene would stick to something else. And it is accountable about the cell replication, and if there is a flaw, it may be transformed into a malign cell. But just forget all about that, because these mechanisms are still being discovered, and doctors don't know how changes in the sequencing would kind of alter the genome. So, but that is also quite important, and of late, machine learning may be of help. We think there are 30,000 genes in the genome, and we can construct a model with the sequence of input letters, around 10 to 30,000 letters, and we supply information about the type of the cells and how they operate and work and what they serve for. And we'll have 30,000 examples. We'll have the different networks and transformers and convolutional networks, and here you can see the most popular models given in the slide, and they can help determine how the intensity of a gene's operation may depend on the sequence of letters, but all of them are fraught with a problem. The point is that genes operate differently in different types of cells, so one and the same gene will activate differently, because the cells may have the same DNA sequence, but they would perform different functions. Well, otherwise ours would not be a multicellular system or body. So, but why not measure the work of genes for each type of cells and try to forecast or predict the changes in the gene's operation if DNA is changed? But there's not a good way out, because only for half of the gene types we have some info available. With respect to the remaining half, we know nothing of that, like what happens if these cells mutate. But it's not that bad. If we look at experimental data, we'll find out that the researchers over the past 20 years have characterized these types of cells. They may have measured another characteristic of genome, like binding between DNA and protein, that correlates with the gene's intensity. There is no straightforward relationship here. And apart from that, we have measured a lot of features of cells, and the most popular features make 40. So we have several types of cells and 40 features, and each type of a cell, this or that feature manifests itself. So, but if we think that these features are correlated between themselves, and can that help us retrieve information on the non-measured characteristics and features with respect to the gene operation? And our suggestion is as follows. We would take a cell type that has two characteristics or features measured, like the gene expression, it's in blue color, and the DNase sensitivity, whether the DNA is free from proteins on this spot. And okay, then let's take and measure like the brain, and the gene expression is not measured there, unmeasured. With the cell type encodings, with the help of cell type encodings, we build a latent manifestation, latent space, and try to restore the characteristics, the features, and in each type, in each case, we should be able to retrieve the characteristics, the features, trying to restore the features as accurately as possible. Do we have many situations when we have two features measured for one type of cells and for another type of cells only one feature was measured? What is the relation between the cell types and features? And this graph shows that there is a good degree of relationship, and we look at the existing non-cell specific structures that receive the DNA, input DNA. Then with the help of transformers they produce a sequence embedding, and that can be broken down into features. And we added one more component, one more pillar. And encoding is transformed into a cell type embedding, and then the characteristics are produced with one part of the neural network. Can that be feasible? Well, we have invented several types of benchmarks, but the metrics we have measured demonstrate that this is feasible. I will not go into detail because of lack of time, and the metrics for each type are comparable with non-cell specific models, so making the model more complex wouldn't reduce the accuracy, and that captures the cell-specific peculiarities of features. The distribution of one of the features of a cell type experimentally measured for two types of cells, K562, the first two lines, and GM12878, which is another type of cells. And peaks stand for DNA sequences with binding proteins. So there are some areas where protein binding patterns are different, and our model can capture and grasp all that. It builds correct embeddings for cell types in latent spaces, and here you can see how that all is treated. Three cell types. It's like the muscle tissue and renal tissue, and we can see how it all is clustered in this or that cell type embedding. If we look at this from a broader perspective, you can see that different cells embeddings are given in kind of gray dots, and the muscle tissue is clustered in one area next to one another, and that may not work for all the cases. Some of the types of cells, like the endocrine cells, are scattered across the embedding space, or we haven't made the final conclusions yet, the final findings. There may be biological deviations when we call or render these cells to one and the same type and call them endocrine. They may not be totally similar from the standpoint of the molecular machines. So is it an artifact, or is it a good representation? We don't know yet. Now we're receiving the first results with respect how we can see the information, extract the information on the gene operation. One deletion or two and three or five deletion of 5 letters inside the gene at 0 point or around the gene, how that impacts the intensity of its work, of the gene's operation. The mutations inside a gene would impact the work, and with DNA there are some segments that are of crucial importance, because mutations in these are of dramatic significance and effect. Like some of the patients have these mutations, and we cannot diagnose them because we don't know whether that will these gene mutations in specific areas of the DNA sequence would make a difference or not. And Olga Kardamon, I would like to thank her especially. Thank you for leading our team, our group. Benjamin, thank you so much for a very curious presentation. Now we have some time left for questions. Like a question, if I may, it is a very interesting and complex topic, and to make it clearer for the lay people, can you tell us what the mutation is from the evolutionary perspective? Because it has its own time scale, and what you analyze, you have kind of your own frame of reference. For me, mutations mean thousands of years. Or am I confusing something? Can you tell me? This is an important topic. Just for those who are not in the know, can you elaborate on that? Yes, thank you for the question. I rejoice I'm on the Zoom, because medical genetics in the audience, in the room, in the hall would throw tomatoes, dropping tomatoes at me. So gene mutation is a slang term that the genetic professionals say variants or invariants. So in my report there are differences between people that surround us and go and see a doctor. And the stories about mutations that either assert themselves in the course of evolution or not is a separate case totally. And you know, with respect to the time span of hundreds of thousands or millions of years, it remains to be seen, but that information will help us judge whether this variant is beneficial, neutral or harmful. But no, I'm talking about variants of a specific individual, and we don't know whether it will entail a kind of disease progress or not. Benjamin, thank you for a detailed answer. Now probably the term mutation is clearer to our audience. I wish you good luck in your undertakings, in your studies. We'll look forward to new results.